Chapter 3, we'll be going over the cell, which is the fundamental unit of life. The cell is considered to be the fundamental unit of life. Every living organism has at least one or more cells that compose it. And the cell is the fundamental unit of life because the cell is the smallest unit that is capable of performing all the activities required for life. So remember back in chapter 1, we talked through some characteristics that all living organisms shared, and the cell is the most basic item that can do those things. So things like subatomic particles, atoms, things that we talked about in chemistry, those don't meet the requirements of what's considered to be alive. So some organisms are only one cell, things like bacteria, maybe some archaea. Those are going to be the single-celled organisms that has the capacity to be alive. You and I are composed of trillions of cells, and so we are made up of, and we begin as one cell, but we are made up of trillions of cells to make up our whole organism as adults, or at least as growing organisms. All cells on Earth can be classified into basically these two domains, and the two domains are how we start the classification system of all life. So domains indicate large groups, and the first domain is prokaryotic, and the second domain is eukaryotic. So prokaryotic domains are considered to be single-celled life. These are your bacteria and archaea. They're going to be really, really simple and basic organisms, and they have been around for roughly 3.5 billion years. So that is an outrageously long time that bacteria have been able to survive and adapt and exist. Kind of a fun fact for you from your book, there are more bacteria living in your mouth right now than the total number of humans who have ever lived. So bacteria is usually so small that we cannot see them with the naked eye, so we use a microscope to be able to see them. Internally, bacteria have no organelles in them. However, they do have DNA. So DNA, as you can see in the picture below on the left, consists of what's called a nucleoid. Sometimes it's in a circle form, sometimes it's in the funky shape you can see on the screen, but all bacteria do have DNA. On the right side is the eukaryotic domain. So in eukaryotic domain, the cells here are larger and much more complex. The difference, additionally, is that the eukaryotic cells are going to have membrane-enclosed organelles. So this means that the organelles that are inside of these cells are going to have some sort of protective membrane around them. They appeared roughly 1.8 billion years ago, and it is believed that they essentially evolved from prokaryotic cells. Eukaryotic cells can exist as a unicellular form, so the organism could be one single cell, or it could be multicellular. So unicellular in this case, they would still be microscopic, so they would be very small. We'd still have to see them with a microscope, but they can live and exist as a single cell organism. And so a question to ask yourself is if you are prokaryotic or eukaryotic. And the answer would be that you are eukaryotic. Here are examples of the two domains and then how they are further subdivided by kingdoms. So the prokaryotic organisms consist of the two bacteria and archaea. Bacteria, as you are probably familiar with, are going to be these very small organisms. They have a wide array of places that they can live in. They can live inside your mouth, on the surface of your skin. They can live on rocks, plants, basically any organism. The archaea group is going to be for the very ancient, or what's considered to be ancient, bacteria forms. But archaea essentially means that they can live in the most extreme of extreme environments, meaning a place that's maybe really acidic, maybe it has the hottest temperature on Earth or the coldest temperature on Earth, in really high salinity, so really salty environments. That's where you're going to find the archaea bacteria. The common kingdoms of eukaryotic organisms are going to be plants, animals, fungi, and protists. So humans fall into the animal category. Fungi, for example, would be things like, of course, your mushrooms, as well as the tiny little fuzz that you sometimes see on your produce as it gets older. It's also going to be yeast. Protists are going to be little organisms that can live on their own. We oftentimes find these protists in bodies of water. And then, of course, you are probably familiar with what plants are. So again, prokaryotic organisms are simple-celled organisms. They have no nucleus, but they still have DNA. So that's a common misconception. Sometimes students get mixed up and think that because prokaryotes have no organelles like your endoplasmic reticulum, mitochondria, chloroplasts, Golgi apparatuses, they think that they don't have any DNA, but they do still in fact have DNA. However, their DNA is not enclosed in a nucleus. Prokaryotic organisms date back at least 3.5 billion years, so they've been around for a very long time, and they have the capacity to survive and adapt much more efficiently and much better than humans. So let's take a look at a traditional bacteria. So here you can see, or what you can see is that all the items that are in blue are items that are unique to prokaryotic cells, which means that the items that are in black are found in potentially eukaryotic cells. So not all eukaryotic cells will have all of them. What it's saying is that it's not just found in prokaryotic organisms. So let's kind of talk through a couple things on the slide. First of all, we have what are called plasmids. So plasmids in bacterium are these circular forms of DNA. To that point, there can also be an additional nucleoid, and a nucleoid is the region inside the bacteria in which you find the DNA. But remember that the DNA is not compartmentalized in what's called a nucleus. So sometimes bacteria will only have plasmids, sometimes they will only have a nucleoid, sometimes they will have both. Then on the surface, there might be what these sticky things that are called pilus or puli. That's going to help the organism stick to a surface. It also helps for communication between bacterium. So usually below the pilus is a structure called the capsule. The capsule again is going to be similar to the pilus in the fact that it can be for attachment or a sticky surface. But additionally, the capsule can act as a protective wall. So in medicine, for example, when you're trying to treat a bacteria with an antibiotic, it has to be able to get through that capsule layer to get rid of the bacteria. So those four items in blue are what can be unique to bacterium or prokaryotic organisms. The other items that are listed here in black, like cytoplasm, cell walls, plasma membrane, ribosomes, flagellum, those can be found in or on eukaryotic organisms. So eukaryotic organisms, compared to prokaryotic organisms, are really large. In fact, it's considered to be roughly about 10-fold bigger than prokaryotic organisms. And also comparatively, they are much more complex because they have lots of internally working organelles in order to help them carry out the cell's functions. So the organelles that are found in eukaryotic organisms perform special or specific functions that allow the cells to do what it's supposed to do. Oftentimes, organelles are called little organs, and similar to your body, that's where the name comes from. Your organs have specific functions that help you as a human being to be a human being. The organelles are like little organs to the cell. So animals and plants contain these organelles, and so for you as a human, as well as in plants, those organelles have a membrane around them, which is what makes you, in many ways, different from prokaryotic organisms. So you'll also remember that prokaryotic organisms do not have organelles. They do still have DNA, but the DNA is not surrounded by a nucleus. Whereas over in this picture of an epithelial cell, you can see that the nucleus is, in this case and in many cases, commonly found dead center in the middle of the cell. And it is enclosed, meaning that it has a membrane around it. So all eukaryotic cells are fairly close and fundamentally alike. All eukaryotic cells are going to share, for sure, a plasma membrane, a nucleus, and mitochondria. So you can see in the picture on the left side, there's a picture of an idealized animal cell, and the picture on the right is a picture of an idealized plant cell. So there's going to be some common structures and organelles that are going to be unique just to animal cells, and then some organelles and structures that will be unique just to plant cells. So your picture down here has the nucleus you can see is shared by them, and then a couple different organelles. So here we can see some mitochondria that are shared. The plasma membrane is found on each of them. And then sometimes they'll have a smooth and rough endoplasmic reticulum. There's some Golgi apparatuses that can be shared amongst them as well. So looking at some more illustrations and pictures with this information on it, on the left side is your standard cell picture of eukaryotic organisms. Up in the corner in pink is what's called the lysosome, and the structure is not actually pink or green, but the lysosome is an organelle that's only found in animal cells, whereas on the right, the items that are in green are items that are only found in plant cells. So chloroplasts, central vacuole, the cell wall, these are items that are only found in plant cells. So therefore, things like the mitochondria, the cytoskeleton, the Golgi apparatus, the plasma membrane, the nuclear envelope, the endoplasmic reticulum, which you'll sometimes see written as ER, ribosomes, those are all shared between the two types of cells. So in animal versus plant cells, you can assume that they have the exact same organelles, except for animal cells have lysosomes and plant cells do not. Also, plant cells have chloroplasts, they have cell walls, and they have a central vacuole. We're going to go through what these structures and organelles do coming up, so we're going to talk more at length about them, but just make sure you know that those items are unique to plant cells. So if you looked underneath a microscope at a cell and you see that the cell has cell walls and chloroplasts, you can automatically know that this has to be in the plant cell category. Animal cells cannot have chloroplasts and they cannot have cell walls. So we'll start talking about cell structures. The first one we'll talk about is the cell membrane. So to survive, every cell must maintain a relatively balanced internal environment as well as the external environment. So the external and internal environment oftentimes have specific names to them, and we can call them some sort of fluid. So inside the cell wall, we will call that typically the ICF, which stands for the intracellular fluid. And then outside of the cell, we call that the extracellular fluid or the ECF. So those two names are what apply to the separation between those two spaces. And what's separating them is the plasma membrane. And this is what compartmentalizes the two. The plasma membrane is ultimately really, really thin, and it's also very flexible. It's a relatively simple structure, but it does a lot of work for maintaining the integrity of the cell. So plasma membranes then are made up of phospholipids, which is a form of a lipid or a fat. It will have some proteins on it that can be integrated, or sometimes they're peripheral, and they oftentimes in animal cells will have a substance called cholesterol. The cholesterol in this case is not the cholesterol that you think about being the kind to be found in your blood that causes you to have high cholesterol. The cholesterol in this case is to help with the membrane maintaining structure and some integrity to it. So to individually look at a phospholipid has particular orientation to it. In a phospholipid, there is a single head, and that head is always going to face the watery environment because the head likes water. It's what we call hydrophilic. Hydro meaning likes, or hydro meaning water, philic means it likes. Whereas the fatty acid tail is hydrophobic, hydro meaning water, phobic meaning adverse to, and it will always face away from the watery environment because it does not like water. It does not want to interact with the water. Here is an illustration of a standardized plasma membrane. You can see here that there's the phospholipid bilayer. You can recognize all the circular heads. So remember, the heads like water, they're hydrophilic, whereas the tails, you can see sort of in the center there, that are represented by the orange lines. Those are hydrophobic, so they do not like interacting with the water. And there is a two-layer system of them. So they are going to have one layer facing the external, or what's called the extracellular fluid, that ECF, and then one layer is going to face the internal, called the intracellular fluid there. And so those two layers combined make up the whole plasma membrane. Then we'll have some proteins that are going to span the entire width of the plasma membrane. Sometimes they're just on the surface, and then there can be some cholesterol that is found in there as well. Membranes do a very effective job in maintaining the passageway of materials. We oftentimes describe your plasma membrane as what's being called selectively permeable, meaning that it will allow some substances to cross, and other times it will prevent substances from crossing. So the most important function is to regulate the movement of those items into or out of the cell. So organelles that have their own internal membranes will have this as well. For example, the nucleus, the endoplasmic reticulum, the Golgi body, those organelles that are inside the cell will also have membranes to help regulate input and output. Remember, membranes and membrane-bound organelles are not found in prokaryotic organisms. For the selectively permeable part of a cell, remember that sometimes substances are free to pass, so they would, for an example, get a nice green light. There are other times that substances are going to be regulated, so there's more of like a yield sign because they could have to be able to pass when conditions are right. And then there are times that certain substances cannot pass at all. So hormones are a good example of this. So if we talk about, for example, the male hormone testosterone, testosterone in men is needed for nearly every cell of the body, but it's needed in various amounts. And so some cells are going to have testosterone flowing freely into them. Others are going to have sort of that yield, meaning that they'll have to regulate it because they don't need as much testosterone. And then there are going to be some cells that don't require testosterone at all. And so that's, they would get like the stop sign. And it's an important thing when we talk about how cells can regulate substances coming into and out of them. So next up is talking about how the transportation occurs. So there are two main divisions of transportation. There's what is called passive transport or active transport. So passive transport requires no energy, therefore the cell doesn't have to use any of its precious energy that it generates to move anything. The important idea here, or the important idea of passive transport, is that everything moves from what's called a high to low, meaning that where you have a lot of a substance on one side, it will move to where there's less or none on the opposite side. There are three main types of passive transport. So we have diffusion, osmosis, and facilitated diffusion, and we'll talk about each one of those in the upcoming slides. The second division then is active transport, and active transport requires energy, and active transport moves items from low to high. So their concentration gradient is just the exact opposite of passive transportation. So passive transportation, here we have diffusion. Diffusion, again, is going to be moving from an area of high concentration to an area of low concentration. So on the top side of the membrane in that picture, we can see that there are lots of tiny little pairs of those red circles, and there's more on the outside than there are on the inside. So the natural motion is going to be moving from an area of a high amount to an area of low amount of them, and that's what the concentration is referring to. So if you think about perfume, perfume functions in the same way in that when you have a spritz of perfume, initially it's very concentrated where you spray it, and then it permeates throughout wherever it's at because it's moving from a high concentration to a low concentration, and with it, it carries the smell of the perfume. So diffusion of water, and water in particular, is called osmosis. So osmosis only refers to the movement of water, and it is a movement of water from high to low concentration. So for example, when you place meat in salt, it will cure it by drawing out the water. So there's more water in the meat, and the salt is going to pull it out, and therefore you can see, like in this example in the picture, on the right, the red circles with the white dots are more concentrated on the inside than on the outside, and so it's going to move from high to low, but in this case, it's only water. So the final form of passive transportation is facilitated diffusion. In facilitated diffusion, oftentimes this one gets confused because it requires help or some sort of adaptation to move across a membrane. So oftentimes people think that this is an active form. This is still diffusion. It's still moving from high to low, but now the molecule has to use a transportation mechanism. In this case, it's called a protein to move across the membrane. So the next category of membrane transportation are active transports. So remember that active transport means that the substances are moving from a low to high concentration. And so the picture on the left is in fact upside down, just show you that the lower to higher concentration is what's happening here inside the cell. So what this means is that in a normal situation, sodium typically moves from a high to low concentration. You can see the high ion concentration on the top and the low ion concentration on the bottom. Therefore, in a normal diffusion situation, sodium moves down, but because this is an active transport, it's going to use energy and it will go against its normal movement. And so in this case, it's going to be pumping sodium ions into the cell against its normal concentration gradient using energy to move it. So exocytosis and endocytosis are two other forms of transportation. In this case, endo means inside, cyte means cell, and exo means outside, cyte means cell, so endocytosis. So this is the importation of substances into the cell. Endocytosis is the importation of substances into the cell, whereas exocytosis is the exportation of substances to go outside. So why would you need this in addition to your passive and active transport? Well, occasionally there are large substances that have to be transported in a large way. So here we have endocytosis, the gathering up of all these purple dots and bringing them into the cell. Whereas on the other side, we have these large yellow circles going out of the cell. They can't be passed through the membrane because they're too big, so this is how they're able to be transported. So now we're going to get at some of the specific organelles and talk about them. So the first one here is going to be the nucleus. So if you remember, the nucleus is only found in eukaryotic organisms, and by far it's typically the most prominent membrane-bound organelle in eukaryotic organisms. Prokaryotic organisms do not contain a nucleus, but they do still have DNA. And remember that eukaryotic cells exist among fungi, plants, animals, and protists. So those four things are made up of eukaryotic cells. So the nucleus, of course, contains a cell's DNA, and the DNA is going to be arranged in structures that we call chromosomes. Chromosomes then contain specific DNA sequence that is unique to every individual, and within the structure of the nucleus, it's also going to have a membrane around it that we call the nuclear envelope, which basically can also be called the nuclear membrane. And then within it, they have these tiny little openings called nuclear pores, and the nuclear pores help the nuclear envelope to allow substances to move in and out of the nucleus. So DNA, again, is organized in what is called a chromosome, and it is wrapped around proteins that we call histones, and that allows it to be very tightly coiled and make the structure called a chromosome. So the chromatin is sometimes what we call the fiber when it is wrapped around a protein and it's more flat, whereas a chromosome is when it twists together to form, kind of like you can see in the picture on the slide. Every human cell contains 46 individual chromosomes, and they do oftentimes look like the picture on the far right when we are able to look at them underneath a microscope. And so that's why we've put the picture on the slide here. So then deep down inside the nucleus is another organelle called the nucleolus. The nucleolus is the location within the nucleus, but this is going to be particularly for producing what is called rRNA, which stands for ribosomal RNA. The ribosomal RNA is an important substance to make the organelle called ribosome, and the ribosome is an organelle that we're going to talk about in just a little bit. One of the activities that the cell is going to be tasked to do using the nucleus as well as other organelles is to make what is called protein. So DNA in the nucleus is going to be a set of instructions for making these substances called proteins. And remember, proteins are one of the 4 macromolecules. Proteins in this case are a package of directions that tells the cell what to do, and occasionally cells need those directions to say, you need to produce more keratin for your skin cells, maybe you need to have more strength in a cell, maybe you need to produce more nail cells. That's what proteins are going to be doing for the cell. So DNA is going to be transcribed into RNA, and then RNA is going to be translated into what eventually turns into a protein. And that's what a main function of the nucleus, along with other organelles, are going to be doing. Several organelles are going to be involved in this process of manufacturing proteins. So it begins in the nucleus, but then from there it's going to be going through an organelle called the endoplasmic reticulum. And on the surface of the endoplasmic reticulum are another organelle that we call ribosomes. And ribosomes, as I talked about when it came to the nucleolus, is what's going to be made from the rRNA found in the nucleolus. So ribosomes are the tiny little purple dots that you can see on the endoplasmic reticulum. They occasionally can be attached to the endoplasmic reticulum, but they can also be found what is called floating around the cell. So we also have the Golgi apparatus, which is another organelle that plays a role in manufacturing proteins by collecting all the proteins, modifying, adapting them, and then moving them to where they need to be in the cell. So cell protein production begins with the transportation process. DNA has to be changed into RNA, and then from there, That happens inside the nucleus, that our RNA molecule will be moving out of it. It will head into the endoplasmic reticulum that has the ribosomes on it. Then from there, it will be translated into what is going to be called a protein. So it goes DNA to RNA via transcription, and then RNA to protein via translation. And then once it gets to the Golgi apparatus, it's going to be sorted and sent to where it needs to go. So two organelles here, after we've got done talking about the nucleus, is the endoplasmic reticulum. So in the endoplasmic reticulum, there are two forms. There's what we call the smooth ER and then the rough ER. Smooth ER can sometimes be abbreviated as the SER, and the rough ER can sometimes be abbreviated as the RER. The difference is that on the surface of the rough ER, it is going to have ribosomes, whereas on the surface of the smooth ER, it's not going to have ribosomes. So the smooth ER's role is to produce what are called lipids. Again, lipids are going to be the fats of the cell. An example of what the smooth ER is going to be producing are going to be steroids. The rough ER that have the ribosomes on them are responsible for producing the proteins that the cell needs. Now, mind you, it is the ribosomes that actually do the activity of producing the proteins. They just get collected by the rough ER. The next organelle will be the Golgi apparatus. And the Golgi apparatus's role is to kind of act as a post office. So it's going to be collecting all the proteins, or basically anything that the cell makes. It will finish off the proteins by adding or adapting or modifying them as necessary. It will then sort the proteins, for example, and then ship them either within the cell or outside the cell, wherever they need to be. So think of a post office and the fact that you collect or give the post office your mail and all forms of packages. The post office is then going to sort them and ship them and deliver your packages and mail where they need to go. So inside the cell is going to be moving anything that the cell produces in tiny little taxi cab-like structures called vesicles. And vesicles allow transportation of these organelles to happen seamlessly, allowing them to be protected as they move about. So the next organelle is a lysosome. So a lysosome is a type of vesicle, and the lysosome is not involved in protein production. And you'll remember that lysosomes are a unique structure or organelle to the animal cell. Plant cells do not have them. So roughly there are about 50 fatal genetic diseases that can be caused by defective lysosomes because they play such a huge role in the cells. So lysocells, you can think of them being like the cleaner. They're there to remove any worn out debris, any organisms such as bacteria, any cellular components that need to be broken down. So their role is pretty much to keep the inside of the cell clean. So mitochondria and chloroplasts are two structures that are going to be powering and supplying the energy for the cells. These two organelles are going to be really, really important to produce the energy the cell needs. Plant cells are going to have chloroplasts and potentially mitochondria, where animal cells are only going to have mitochondria. So chloroplasts and mitochondria can be found in plant cells. So first, let's talk about chloroplasts. Chloroplasts are found in all plant cells and the cells of algae. They are so important to carry out the process called photosynthesis. At the most basic level, photosynthesis is essentially using carbon dioxide and water to produce sugar. Sugar gets stored up in the plant, and then we eat the plant. So that's what's going to give us our energy by getting that sugar. So photosynthesizers are in bacterium and algae and plants. They've been around for many billions of years before humans were ever around. Chloroplasts are green in structure, which is why we often, whenever we see them in plants, that's why we see green. And they have to harness and use the sun to be able to produce the energy that, again, gets stored in plants. So here you can see a picture of a cell or a plant cell, and the green blobs are the chloroplasts. So then we have mitochondria. So the mitochondria provides energy to nearly all eukaryotic cells, including our own. Within the mitochondria, there's a series of enzymes that perform a chemical reaction that we call cellular respiration. Cellular respiration uses oxygen to create and harness the energy that we use from processing sugar. And where do we get our sugar from? Well, we get sugar from the items that we eat, in particular things that are going to be going through photosynthesis. So energy is then stored in the form called ATP. And then ATP is then used to power many cellular processes. So you can think of ATP as like the gasoline to your car. So in order for your car to go, you have to have energy, and that's exactly what ATP is to the cell. So a vacuole is going to be a structure that is only going to be found in plant cells, oftentimes in the very center of these plant cells. Vacuoles and central vacuoles in plants can be used for things like food, nutrients, water, or pigments, and it's very, very commonly found right in the middle of those large plant cells. Humans and eukaryotic cells can have vacuoles, they're just not considered to be central. So here on the right side, just want to make sure you note that if the plant has a central vacuole, usually it's full of water, but when fungi and protists, which are eukaryotic organisms, also have a vacuole, in this case, the vacuole might be for storage, for anything that the cell needs, for nutrients, for pigments. That's what it's used for. So then we have two propeller structures and appendages that oftentimes extend off the cell. So if there's multiple of them, we call them cilia. If there's usually one or maybe a longer one, we call them flagellum. So flagella is a long extension that comes from a cell. A great example in humans is sperm. That sperm is going to contain a long flagellum to work its way to meet a human egg in order for fertilization to take place. Cilia look kind of like shag carpet on the surface of the cell, and cilia are usually short and numerous, and their role is to create motion in order to move substances in a direction. For example, along your trachea, you have lots of ciliated cells to get out the debris and phlegm that you breathe when you breathe in air. So then we have a structure called the cell wall. So in the cells of plants, fungi, and some protists, they sometimes have a cell wall. I want to make sure that there's big emphasis that this is not the same thing as a cell membrane. A cell membrane is also called the plasma membrane. That's something entirely different from a cell wall. A cell wall is made from fibers of a carbohydrate that we call cellulose, and that creates more of a rigidity, creating the plant to look a little bit more square, sometimes a little bit more oval. But cellulose is what's found in cell walls, and human and animal cells do not have it, and therefore it's not the same thing as a cell membrane.